Welcome back, everyone. This lecture, we're going to talk about the process of digestion and absorption. They're better talked about together rather than separating each of them. And when we discuss this, we're going to start with carbohydrates, then we're going to move to proteins, and then we'll end our discussion on lipids, as well as some of the vitamins and minerals within our diet. So physical digestion is really the first part of the process of breaking down food, and it begins with chewing. So the first thing is there's mechanical digestion by teeth. This creates a smooth, softened mass of food that passes very easily from the mouth to the pharynx, which is the back of the throat, to the esophagus, and eventually into the stomach. The process of swallowing moves the food from the mouth to the stomach via the esophagus, and this process is known as deglutition. And there's a specific process called the swallowing reflex that occurs during swallowing. So the first thing that happens, here's the bolus of food. The tongue pushes the bolus of food against the soft palate. So this is the soft palate. It's a non-rigid part of the roof of the mouth. There's the hard palate as well that's made up of one of the bones of the jaw, and there is the soft palate. So it pushes the bolus of food against the soft palate to the back of the mouth, and this is what triggers the swallowing reflex. Now, if you notice here, if you look at the back of the throat, there is where the food is going is the esophagus, and there's a tube in front of it, and that's our windpipe. So that's the trachea, where air is supposed to go in. Very bad thing, obviously, if food goes into that area. So there's a little tiny flap of cartilage, known as the epiglottis, that folds over top of the airway so that the food passes directly behind it, and it prevents it from going in. So breathing is completely inhibited as the bolus passes along this area. So you can't breathe, the windpipe is closed, trachea is closed, and the bolus passes into the esophagus. And there's a sphincter at the beginning of the esophagus called the upper esophageal sphincter that relaxes and allows for the bolus of food to move through. And then the bolus of food follows a downward trajectory along the esophagus, and this is governed by peristaltic waves. So that's the first thing that helps the bolus of food travel down. It's also aided a little bit by gravity, but gravity is it's not a very big player in the process of a bolus of food moving from the esophagus into the stomach. You can eat food upside down and it'll still go into your stomach. So just before the stomach, there is another sphincter called the lower esophageal sphincter. This must relax in order to allow for the bolus of food to pass into the stomach. So we'll start our process of chemical digestion by talking about carbohydrate digestion. We're going to talk about the absorption of these nutrients at the same time. Absorption of carbohydrates is limited only to the small intestine. And the other thing that you need to know is that carbohydrates can only be absorbed as monosaccharides. Anything bigger than a monosaccharide passes right through. We don't get the nutrition from it. And so where do we get carbohydrates in our diet? Well, there is in the form of cellulose. Well, we can't break down cellulose. And cellulose is really just a whole bunch of glucose molecules that are linked together with a flipped, it's a flipped bond. It's called a 1,4 beta ether linkage. If you're interested, you don't need to know that. But we don't have an enzyme that can actually break down the cellulose into individual glucose molecules. And so it passes right through. It's generally considered a type of fiber or roughage in the diet. There are complex carbohydrates that we get from starch containing compounds, rice, potatoes, some animal complex carbohydrates in the form of glycogen, Um, and then we also get our carbohydrates in the form of disaccharides, so maltose, sucrose, and lactose. So enzymes that help with the digestion of carbohydrates starts out with amylase. And amylase is an enzyme that helps break down starch and glycogen into glucose monomers and maltose. So two glucose monomers linked together. We get amylase in two areas. First, we get amylase from our salivary glands. So there's salivary amylase, and then there is pancreatic amylase. The salivary amylase, um, it will start the process of breaking down starches. So I don't know if you've ever done the experiment where you take a little tiny piece of potato and you chew on it for a really long time. It'll eventually start to taste sweet because the amylase is breaking down the little piece of potato. Um, it's one of those experiments that I did in, I think it was like grade 11 biology or something, and it was to demonstrate the presence of amylase in salivary glands. Anyway, so that's the beginning part of the digestion of carbohydrates. Also within our GI tract, we have the ability to break down the disaccharides. And we have to have this ability because we cannot absorb molecules in the form of disaccharides or sugars in the form of disaccharides. They have to be in monosaccharide form. So these disaccharidases, disaccharide, and it breaks it down. So it's the suffix ase is on it. We have, and these are really easy to remember because maltose is broken down by maltase, sucrose is broken down by sucrase, and lactose is broken down by lactase. And we find all of these disaccaridases on the brush border of the intestine. We're going to talk about what the brush border is. So not to worry, we're going to get to that point. And this helps break down those disaccharides into monosaccharides so that they can be absorbed. Brush border. So if we look at the small intestine, what we'll see is this would be like the lumen here. There would be the lumen and there would be all of these villi that are folds that are increasing the surface area of the small intestine. So lumen here, this is where all the food stuff is. And then if you remember the histology, there's the epithelial cells that line the entire small intestine. And then if you take one of those epithelial cells, and these particular cells, they have a specific name. They're called enterocytes. So entero means intestines, cyte means cell. So these are the intestinal cells. If you look very closely at one of these enterocytes, you'll see that there are all of these little microfolds. So there's folds on folds. And these little folds, they kind of look like the villi that you'd see in the small intestine, but they're really tiny. And it almost looks like a brush. So they call it the brush border, and they call these little folds microvilli. This is the location of these disaccaridases: maltase, sucrase, lactase. And here's how carbohydrates are digested. So the first thing that happens is you ingest something that contains starch or glycogen. Salivary amylase is going to start breaking that down, and it's going to start breaking it down into either glucose monomers, so there should be a line that goes to glucose as well, or it breaks it down into those maltose disaccharides. So those maltose disaccharides. When you're in the small intestine, these disaccharides are all present in the small intestine. They travel along the small intestine, and when they are up against the brush border of the small intestine, they come into contact with maltase, sucrase, and lactase. The maltose is broken down by maltase into two glucose monomers. Remember, only monosaccharides can be absorbed, so this is good. We can absorb these. Sucrose is broken down by sucrase, and that gives you one glucose molecule. and one fructose molecule per sucrose molecule. And then lactose is broken down by lactase, and that will give you one glucose molecule and one galactose molecule. Now we're in this stage where we have broken everything down into monosaccharides, and these are now absorbable forms of carbohydrates. One thing to note is that this amylase, while it comes from the salivary glands, it also comes from the pancreas. So you have pancreatic amylase as well that will get put into the small intestine. So any kind of starch or glycogen that wasn't fully broken down will continue to get broken down when it's in the small intestine as well. So when we think about the absorption of these monosaccharides, we're going to talk about each of the three that we were left with. We were left with a bunch of glucose, a bunch of fructose, and a bunch of galactose. And we'll talk about each one. Glucose and galactose are identical. The process of absorption is identical. From the transporters to the ions that are required, everything is the same. Fructose is a bit of the oddball out. So when we think about the absorption of glucose, we are in the lumen of the small intestine, okay? And we're first going to talk about glucose. So imagine that there's a bunch of glucose in the small intestine here. When you think absorption of glucose, think sodium dependent. Think it needs sodium. And it's absorbed into the intestinal cell from the lumen into this intestinal cell or the enterocyte by a specific transporter that you should actually have heard the name of before by now, is that SGLT, sodium glucose linked transporter. And that is, if you remember, secondary active transport, okay? And so you bring the sodium across and you bring the glucose across at the exact same time. And they're transported across the cell membrane into the enterocyte. Once they're in their enterocyte, the glucose is further transported across the cell and eventually binds onto a specific transporter called a GLUT2 transporter. And this GLUT2 transporter, any of the GLUT transporters, there's a whole bunch of different GLUT transporters, but they all have a similar function in transporting carbohydrates. These ones are specifically glucose. And so you have the GLUT2 transporter specifically transports glucose, and it brings it out of the cell. That glucose can then be taken up into the capillary, and it enters the bloodstream, and it can be transported to the brain. It could be transported to muscle cells. It could be transported and stay in the liver. It gets transported to all the cells of the body. If you remember from our discussion, I think it was the second class, we talked about SGLT being a secondary active transporter. So it's very important that sodium is traveling down its concentration gradient from high concentration to low concentration, and it pulls in glucose against its concentration gradient. And so the point here is that this low sodium concentration within the enterocyte is maintained by the sodium-potassium pump. And so while this process requires energy, it's an indirect form of energy because the process that's actually transporting the molecule does not itself require the energy. So this is the secondary active transport mechanism of the absorption of glucose. Galactose, we don't have to talk about anything because it's an identical process. Whatever we talked about with glucose, just sub in the word galactose and it is exactly the same. Fructose is a little bit different. Fructose does not depend on sodium in order to be absorbed in the GI tract. So the absorption of fructose uses a specific transporter called GLUT5. And so imagine fructose, after it gets broken down, after sucrose gets broken down into glucose plus fructose, that fructose molecule is going to bind on to the enterocyte, and it's going to bind on a specific transporter called a GLUT5 transporter. Once it's inside of the enterocyte, that fructose travels across the enterocyte and eventually binds on to a GLUT2 transporter, the same one that transported the glucose and the galactose. That fructose is then brought into the bloodstream and it can be transported to the cells for energy. So we'll talk a little bit about lactose intolerance. It's a very common condition whereby one of the intestinal brush border disaccharidases is missing. Either you don't have it or you don't have enough of it. It's a very common condition that usually develops in adulthood. The majority of adults have some element of lactose intolerance. You probably didn't have lactose intolerance as a kid, but you actually may have developed lactose intolerance a little bit later, either at the end of your adolescent period or mid-adolescent period, or some of you will probably go on to have lactose intolerance later on. Lactose intolerance is not an allergy to lactose. It just means that you are deficient in the enzyme to break down lactose. So having a deficiency in this enzyme leads to undigested lactose. Remember, lactose is a disaccharide. It cannot be absorbed as a disaccharide. And so you can imagine that as this lactose travels through the small intestine, it stays as lactose. It stays as a sugar. And it passes through your small intestine as a sugar, as a disaccharide. It goes onto your large intestine as a disaccharide. And sugars that don't get absorbed or can't be absorbed, they tend to draw in a lot of water into the intestine in an attempt to dilute it. And so with more water in the intestine, it could actually lead to diarrhea. Part of the microorganisms that live in your large intestine, they can actually start to digest the lactose. The problem with that is that you can't absorb it. There's nothing to absorb the sugars that are produced from the breakdown of lactose, but those microorganisms also create a lot of gaseous compounds. So people with lactose intolerance tend to get a lot of bloating as well. And so this, as I mentioned, the lactose gets broken down, leads to a lot of bloating. People get abdominal pain. There's discomfort. You can have diarrhea. It's not a pleasant experience to have lactose intolerance. So people will generally, if they have lactose intolerance and they're going to eat or drink something that contains lactose, they'll take Lactaid or they'll take some sort of a pill that gives them the lactose enzyme. That helps them break down the lactose. It's a little bit easier to just buy lactose-free things. So here's an interesting question. There's a way to diagnose lactose deficiency. So using your understanding of the digestion of lactose and the absorption of its monomer units, so the glucose into lactose, what type of change in blood glucose would you expect after ingesting lactose in a person with lactase deficiency, and B, in a person with normal lactase levels? Just going to change my screen here to my little whiteboard and I'll draw a couple of scenarios for you. Okay, so the first question, well, actually, we'll use the person with a normal lactase. So imagine here that you're testing someone, or I'm testing someone, or someone's testing someone for lactase deficiency. Well, you're going to get them to fast, and that's going to get them into a blood sugar level or blood glucose level that would be in a certain range. It doesn't really matter what the range is. But after their period of fasting, they're going to ingest a particular amount of lactose. So the first thing they'll ingest lactose. And so here's what is going to happen. So you're in the small intestine. And this is the lumen of the small intestine. And there's a whole bunch of lactose that's coming into the small intestine. This is lactose. And that lactose needs to be broken down. So at the brush border, so this would be all of those, I can't really draw microvilli because they'd be too small, but just imagine that there's a whole bunch of microvilli along the lining of the small intestine. And imagine here that there's also a capillary that's kind of running across. So there's blood in there, and so that's our capillary. So that's where the bloodstream is. So this lactose, at the brush border, you're going to have all your disaccharidases. So you're going to have sucrase, you're going to have maltase, and you're going to have lactase. So as this lactose comes up to the lactase enzyme, that lactose is going to be broken down into glucose plus galactose. And so you're going to be, somebody with normal lactase function is going to be able to break down the lactose, and they're going to break it down into glucose. And that glucose, however much glucose there is from all the lactose molecules that you're digesting or breaking down, that glucose is going to be absorbed into the bloodstream. So overall, you'll have increased glucose in the blood. And so if you were to check somebody's blood glucose, say an hour or 2 hours after ingesting lactose, so number 2, you check blood glucose. 1 or 2 hours after, you'll pick up the presence of all of that glucose that's being absorbed. So what you'll find is that there'll be an increase in blood glucose if the person, they have normal, a little N with a circle around it stands for normal, if they have normal lactase function. So if somebody has suspected, there's actually better tests than this that are actually done, but this is one of the possibilities. So you send someone to ingest lactose after a period of fasting. They ingest it, they break it down into glucose. You check the blood sugar level later. Well, the only source of glucose you might have a little bit from the liver because of fasting, although you've now eaten lactose. The only source of that glucose is going to be from the breakdown of lactose if you have broken down lactose. So that would be if somebody has normal lactase function. If somebody has deficient lactase or no lactase, I guess we'll go with the no lactase. Well, you have your same small intestine. Here's the lumen, and you've ingested lactose. Well, this lactose is going to come up across the brush border of the intestine. The brush borders on really around the entire circumference, so it doesn't matter what side I draw it on. So this lactose is going to brush up against the brush border. The problem is there is no lactase. So you can't break it down. And so it stays as lactose. Well, you cannot absorb lactose because it's a disaccharide. Cannot absorb it. And if you can't absorb it, if you were to check the person's blood sugar an hour or two after ingesting that lactose, there would be no glucose being absorbed because no lactose is being broken down. And B, so their blood sugar won't change, won't be elevated. Okay, that's one way of using physiology and using the process of digestion in order to help you understand how someone might go about diagnosing lactase deficiency. Again, this is not really a method that's used very often. It is sometimes, but there are better methods. But this is just the one that best highlights what we've talked about. Okay, so now that we've gone through that, we can actually turn our discussion to the digestion of proteins and absorption of proteins. So we are going to discuss that. The first thing that we need to talk about is how are proteins digested. And so the first thing is that there are some enzymes for protein digestion. And we classify these enzymes in two groups. There are these things called endopeptidases, so enzyme that breaks down peptide bonds. And endo means within. So if you were to imagine a long protein chain, an endopeptidase would break down that protein chain from somewhere in the middle of the chain into smaller pieces because it's within, endo. An exopeptidase breaks off single amino acids from the end of a peptide, from a long protein. It's on the periphery. It's either from one end or the other end. It doesn't break it into small chains. It just breaks single amino acids off. Endopeptidases are often called proteases. And so you can see here that an endopeptidase, if you've heard of some of them before, there's one called pepsin in the stomach. There's a couple of them that come from the pancreas called trypsin and chymotrypsin. You've probably heard of pepsin. You may not have heard of trypsin and chymotrypsin. We're going to talk about all of them. But these are endopeptidases. So you can see that the endopeptidase acts on this protein. So this is a protein with long chain of amino acids. There's an amino end and there's a carboxyl end. And it works in the center and it breaks this down into two smaller chains. And again, examples are pepsin from the stomach, trypsin and chymotrypsin from the pancreas. Exopeptidases, unlike the endopeptidases that would break down this chain into two smaller ones, the exopeptidases work by breaking off single amino acids. And these are really logically named. So there are aminopeptidases, and as its name implies, it breaks off a single amino acid from the amino terminal of the protein. And then there are also carboxypeptidases that break off a single amino acid from the carboxyl terminal of the protein. Most, or really all of these proteases and enzymes that break down protein, they're all released in inactive form or in as zymogens. And this is again so that they don't digest the cells that make them. And so here is the process. The majority of these enzymes to break down proteins, even to break down fats and phospholipids, a lot of these enzymes come from the pancreas. And so there's a process by which the pancreas releases these enzymes, and there's actually a process by which the enzymes get activated. So here is the lumen of the small intestine. And what you'll see is that there is an opening at the top of this diagram where it says pancreatic duct. That's where all the enzymes are coming in, that number 1. So your pancreas is making a bunch of secretions. They're entering the small intestine through that pancreatic duct, but they're released as inactive zymogens into the lumen. So right now they can't do anything here. They can't do anything here. And you'll see that all the zymogens are released, and you'll see what their names are. You'll see there's this trypsinogen. Ogen often means that it's an inactive enzyme. And we talked about this in an earlier lecture, that suffix ogen means it's inactive. And you'll see under that title of zymogens, chymotrypsin ogen. There's one other nomenclature thing that you should know. If an enzyme starts with the prefix pro, it's also a zymogen. So pro-carboxypeptidase, pro-colipase, pro-phospholipase. Every one of these is an inactive form. So the real question is, how do we get all of these enzymes from inactive form to active form? And the way that we do that is the trypsinogen that's released. Trypsinogen travels into the lumen of the small intestine, and it actually comes into contact with another brush border enzyme. This is the same brush border that contained the disaccharidases. It contains this enzyme called enteropeptidase. And so this particular enteropeptidase, it converts trypsinogen into trypsin, so it activates it. And then once trypsin is activated, trypsin can activate all of the other zymogens. And so trypsin cleaves or activates chymotrypsinogen, and it turns into chymotrypsin. Trypsin activates procarboxypeptidase, and it turns into carboxypeptidase. And same thing with prolipase and prophospholipase. They turn into colipase and phospholipase. So now all of these enzymes that are activated can start breaking down what they need to break down. And so you're now in this area of your GI tract where you've activated the enzymes at a time where they're not really going to have as much contact with or they're not going to have as much of an opportunity to break down your own cells. So it'll only break down the food stuff that's in the small intestine, the lumen of the small intestine. So just in case, there's also another little thing that actually gets secreted in this whole slew of pancreatic inactive enzymes, just to make sure that trypsin doesn't become activated a little bit too early. There's a trypsin inhibitor that is secreted in by the pancreatic cells. So all of these zymogens contain a little bit of trypsin inhibitor. On the off chance that there's a little bit of trypsin that is activated, that trypsin inhibitor will be able to stop that trypsin from activating all the enzymes. There's only a little bit that's actually produced. So in the small intestine, when you start activating all of your trypsin, the trypsin inhibitor there's such small amounts that it becomes overwhelmed, and it can't inhibit the trypsin that's produced. So it's just a safety net. there's a trypsin inhibitor as a safety net so that you don't activate all of these enzymes in the pancreas. That would be a very bad thing. And there is a condition that happens when you actually activate all of the enzymes within the pancreas. And it's a condition called pancreatitis. It's whenever you see a term that ends in itis, it just stands for inflammation. And then whatever comes before it is what is inflamed. So the pancreas is inflamed, pancreatitis. Extremely painful, absolutely painful. This diagram, again, is not meant for you to memorize this. I don't care if you know all of this stuff. It's just to show what happens when some of the enzymes get activated early. There's lots of causes of pancreatitis. And if you look at the way that the ductal system and the duodenum are situated, you'll see that here's the left and right hepatic duct that carries bile. This is the cystic duct that carries bile. Here's the gallbladder. And that bile goes through the common bile duct and eventually reaches the small intestine. And notice that the pancreatic duct links up with this common bile duct before it enters the small intestine. So one of the reasons that somebody gets pancreatitis is that this area where the pancreatic duct is becomes blocked. And so what can block it? Most times, it's actually a small gallstone, so a gallbladder stone, a small one that can travel along the cystic duct through the common bile duct and get stuck right at where the junction of where the common bile duct meets the small intestine. That stone gets stuck, and it actually stops not only bile from going through, but it also stops all the pancreatic juices from flowing. And so, again, gallstones are the most common cause, but it's usually, if a gallstone gets stuck right at the junction of where the gallbladder and the cystic duct start, it doesn't matter. You're not going to block the pancreatic duct, but it's when a tiny stone gets stuck right where the small intestine meets, right at that sphincter of Oddi, if you remember that term, sphincter of Oddi, that will block the flow of pancreatic enzymes. And so what will end up happening is your pancreas will keep making these enzymes, and it's just going to back everything up. And everything gets backed up into the pancreas. And eventually, there's so many enzymes that are within your pancreas. So just imagine that there's a pancreas here. There's so many enzymes that there's a funny thing that happens when you have a high number of enzymes within the pancreas. That inactive trypsinogen that normally would have had to be activated in the small intestine by that enteropeptidase, that trypsinogen, it can actually activate itself. If there's high enough quantities, it eventually overcomes the trypsin inhibitor, and that trypsin that's produced and activates all the other enzymes, and your pancreas starts to digest itself, which is extremely painful, as you can imagine. So it's a very interesting condition. It really plays on this idea that you need to make sure that you activate enzymes in the appropriate place. And if you don't activate the enzymes in the appropriate place, you end up with disease processes. Again, I'm not going to ask you about exactly how pancreatitis happens. That's not the point of this. It's really to highlight that the physiology that I talked about where these proteases are activated, it's a very important piece of physiology that has practical clinical application. Okay, so now that we know how proteins are digested, we now have to talk about how proteins are absorbed. And so proteins can actually be absorbed in a bunch of formats. You can either absorb free amino acids, or FAAs. You can absorb dipeptides. You can absorb tripeptides. You can actually even absorb some smaller peptide chains that are more than 3 amino acids long. So anyways, now that now we'll talk about each of these absorptive processes. So when we think, we're going to talk about free amino acids first. And when you think free amino acid, think sodium, single amino acid sodium, single amino acid sodium. It is dependent on sodium. And so here is the lumen of the small intestine. Proteins get broken down into smaller peptide chains or free amino acids. So you're either breaking it down into di and tripeptides, you're breaking these peptides into single amino acids, and some of the breakdown is going to be in the form of these small peptide chains, anywhere between 4 and 10-ish amino acids. We'll focus on the middle one right now, the single amino acids, and it's transported along with sodium. This is very kind of similar to glucose. You transport glucose with sodium, you're transporting single amino acids with sodium. So the first thing that will happen, I guess we'll chat about the single amino acids, is that the single amino acid is transported on a co-transporter. So it's an amino acid sodium co-transporter. They both get brought into the cell, and then these single amino acids travel across the cell over to the side of the cell that has the blood vessel, and you end up with the amino acid being absorbed into the capillary. And that is in exchange, so that when the amino acid is removed from the cell and absorbed into the bloodstream, a sodium ion is brought in. So co-transported to go from the lumen into the cell, so co-transported with sodium. And then there's the antiport system that takes in an amino acid, or brings an amino acid out of the cell, and sodium goes into the cell. But both are sodium dependent. The di and tripeptides are co-transported with a proton. So again, a positively charged ion, but not sodium. And so you can see there are some chains of 3 amino acids long. There are these dipeptides as well with 2 amino acids. And the first thing that happens is that sodium, or not sodium, that di or tripeptide is going to be brought into the cell with a proton. So those di and tripeptides are brought into the cell with a proton. And now there's one of 2 things that can happen. Either that di and tripeptide can be broken down further in the cytoplasm of the cell by cellular peptidases, things that will break down peptide chains. And so if that happens, you end up with the breakdown of the di or tripeptide into free amino acids that then follow the single amino acid pathway with sodium. Or that di and tripeptide can actually just go across the cell and be transported across into the bloodstream in exchange for a proton. So again, when you think of di and tripeptides, it is dependent on a proton, both at the luminal side, so the side with the lumen, and the side of the cell with the capillary. Both of them have a proton associated with their transport. So we've already talked about the single amino acids and the absorption of them. So we're just, if you want to go through that again, I encourage you to read that slide. The larger peptide chains, anywhere between 4 and 10-ish amino acids, they can actually be brought into the cell And there's no transporter. They can actually be brought into the cell via a direct process called transcytosis. And this transcytosis process helps these proteins, they really transverse the cell. They go right across the cell and they end up getting absorbed right into the bloodstream. Interestingly enough, having these big protein chains being brought in without any kind of digestion, you can imagine that if this big protein chain is brought into the bloodstream and it's from food, these big proteins it might cause there to be a problem with the immune system. The immune system might recognize these long protein chains as being foreign. And if these long protein chains are seen as being foreign, there's the potential for allergies to be produced. And an allergy is really just an inappropriate immune reaction to something that isn't a threat. So while it's not defined as this is the cause of allergies, it's thought to maybe play a role, is the transcytosis of small peptide chains that are undigested and brought right into the bloodstream. So we'll now move to the digestion and absorption of fats. These are a little bit more complicated. Digestion and absorption of fats, the reason for this is that fats aren't water soluble. So there's not as much surface area exposed for digestion. So we need a little bit of help with this. So bile salts are help. Bile salts will help break down larger fat globules into smaller pieces to maximize the surface area that we have available for digestion to occur. Bile salts, the reason why they work is that they are something called amphipathic. They have both a hydrophobic region that is in contact with the fat and a hydrophilic region that's in contact with water. And this helps create a stable emulsion of the water soluble component of fat droplets. So it makes it a little bit more water soluble so that there's more option for digestion to occur. And so here's a bile salt. So bile salts are, it's bile salt that's coating a lipid droplet. So we have this fat droplet, which is this large yellow droplet. And then all around the outside, we have these smaller bile salts. And you can see these green dots. Those green dots are the hydrophilic region. So these are water soluble areas, and you can see that they're all facing the outside, which is where the water component would be. And they also have a lipid soluble section or a hydrophobic section that they are all in contact with the lipids. And so the bile salts really just surround lipid droplets and try to break them into smaller bits so that more is available. The way that we break down fats is there's a couple of things that need to happen. Number one, the most common type of fat that we're going to see in our diet are triglycerides. And triglycerides, so it's that glycerol with 3 fatty acid chains attached to it. Triglycerides are broken down by the enzyme lipase, so breakdown of lipids. The bile salt coating of the fat emulsion actually makes lipase completely impenetrable. So what we see here, you can't get lipase to the center to actually break down the fat. And that's a problem. So the pancreas secretes this enzyme that helps. And just like a co-pilot is sort of helping the pilot, there's an enzyme called colipase that is going to help the lipase gain access to the lipid droplets. And so what colipase does is it helps displace some of the bile salts to allow the access of lipase to the lipid core. There's another enzyme that helps break down another type of fat, which is very common. So phospholipids of cell membranes. These phospholipids are broken down by an enzyme called phospholipase. So it breaks down phospholipids, and it's an enzyme. So phospholipase, completely boring name for an enzyme. Cholesterol is also a fat-soluble substance, and this free cholesterol is just absorbed. There's no enzyme that's required for cholesterol absorption. So we'll talk about how lipase works. So lipases break down these triglycerides. So here's glycerol is the yellow part, which is kind of funny. I don't know why they did that, because it's always been the fat has always been the fatty acid chain has always been the yellow aspect, but that's fine. The glycerol aspect is this yellow box now, and these little corrugated lines, squiggly lines, those are the fatty acid chains. And so there's 3 of them in triglycerides: 1, 2, 3. And so just imagine that this triglyceride is within one of those lipid droplets. Lipase and colipase are going to be released, and they're going to, the colipase is going to allow the lipase to have access to the lipid core, and lipase will actually break down this triglyceride into one monoglyceride, so one glycerol unit with a fatty acid chain attached, and 2 free fatty acids. So don't tell me on an exam that lipase breaks down a triglyceride into glycerol and 3 free fatty acids. Doesn't happen. It only breaks off 2 of those free fatty acids. 1 of them stays attached. So lipase digests a triglyceride into a monoglyceride with 2 free fatty acids. Okay. Some of your people that you've come across in the past may have told you otherwise. This class is telling you monoglyceride plus 2 free fatty acids. So the process of fat digestion continues, and you have these lipid droplets that contain phospholipids, you have the lipid droplets that contain triglycerides, cholesterol, and this digestion continues, and you make this thing called a micelle. And my cells are better than your cells anyways. They're sort of partially digested fat droplets. So you can see that in this micelle, there's a bunch of phospholipids, there's a bunch of bile salts, there's a monoglyceride, a diglyceride. Either way, all of the polar regions are pointing to the outside in a micelle, and all the non-polar regions are pointing to the inside of the micelle. So micelles really contain free fatty acids. So you can see the free fatty acids in the center. They contain monoglycerides. So I'm circling a monoglyceride here. They contain diglycerides. I'm circling a diglyceride. They contain phospholipids, and they contain bile salts. And so this process continues and continues. And so here, and it continues until you have all of the products of fat digestion that you possibly can. And so we're going to go through what happens after you digest all of these fats. So we're going to orient ourselves. So here are these blue cells. These are the enterocytes of the small intestine. On the left side, this is the lumen of the small intestine. On the right side, these are where the vessels are. So there's a capillary, and there's actually a lymphatic vessel as well. The lymphatic vessel is actually quite important in this absorption of fats. So the first thing that happens is that you ingest something with fat. So fat containing droplets enter the small intestine, and they're in these large fat droplets. Much too large for there to be any kind of good digestion of these fats. Well, that's okay, because the presence of fat in the small intestine is going to cause there to be some bile salts that are released. And so these bile salts are released, and they come from bile. And these bile salts will coat all of these lipid droplets and break them down into smaller pieces. And so it actually emulsifies them. So you can see these bile salts are surrounding these smaller lipid droplets. Now that you have the bile salts surrounding the lipid droplets, there's those pancreatic enzymes that are also in the mix in the small intestine. So pancreatic lipase and colipase will work on these emulsified fat droplets, and it'll break down those triglycerides into monoglycerides and free fatty acids. And so as this process of digestion continues, you get those micelles with those digested products of fats. Okay. The third thing that happens is that you are going to have some of those products of the micelles be moved out of the micelles and into the enterocyte. So you have the free fatty acids and you have the monoglycerides that are going to be brought out of the micelle, and they're going to be absorbed into the enterocyte. And this happens via simple diffusion. These are non-polar molecules. Non-polar molecules really travel very well through the cell membrane. And you'll see what happens. You'll see that immediately these free fatty acids and the monoglyceride, they are brought to the endoplasmic reticulum. And here's where it's kind of funny. The endoplasmic reticulum actually takes these broken down pieces of triglyceride, and it just repackages them into a triglyceride. So the question is, why did you break it down in the first place? Why did you break it down if you're just going to repackage it and put it into another triglyceride? The reason for that is that triglycerides can't diffuse across the cell membrane. They have to be broken down. So it seems like a wasted step to break it down only to repackage it, but they cannot be absorbed across. The cholesterol component of the micelles is transported into the cell. And so now you have the triglyceride component, you have the cholesterol component, and together, these get packaged with some proteins within the cell. And so together you have cholesterol, you have triglycerides, and you get protein. And these are packaged into these well-packaged molecules called chylomicrons that contain cholesterol, triglycerides, and protein. They get further packaged in the Golgi apparatus, and the Golgi apparatus helps bring each chylomicron to the other end of the cell where the capillary is, so that it can actually be taken out of the cell. And these chylomicrons would love to enter the bloodstream, but they are far too large. They cannot go through a capillary. Capillaries are tiny, these are huge. And so the only place that these chylomicrons can actually get absorbed is in the lymphatic system. The lymphatic vessels are larger. They have larger pores to allow for these chylomicrons to enter the lymphatics. The lymphatic system eventually drains into the bloodstream. So these chylomicrons will eventually enter the bloodstream. So they travel through the entire lymphatic system, and there is a place where the lymphatics drains in. So it still enters the bloodstream, but it just has to take an alternate route because chylomicrons are far too big to enter the bloodstream directly. This diagram seems a bit confusing. I'm only going to tell you what I want you to know about it. There's a lot more physiology and biochemistry here that's not even listed on this page. But there's only a few things that I want you to know. And the few things are actually written at the bottom. So the chylomicrons have entered the lymph, and eventually they enter the bloodstream. So this little CM stands for chylomicron. And that chylomicron in the bloodstream comes into contact with an enzyme that is present. That enzyme is called LPL. And if you remember, the chylomicron contains cholesterol, triglycerides, and proteins. And so this LPL stands for lipoprotein protein, lipase. So the protein component and the lipid component are going to be broken down by an enzyme. And what you get is you get the triglyceride component of the chylomicron gets broken down into free fatty acids and glycerol. So lipase, if it's just the protein, if it's just the enzyme lipase works in the small intestine. Lipoprotein lipase acts in the blood. They're two different enzymes and they have two different products of digestion. So if you see here, the product of lipoprotein lipase is a free fatty acid or free fatty acids and glycerol. Eventually, these free fatty acids and glycerol are brought into the cells to either be used for energy. So here, most cells are going to use free fatty acids for energy, or if there's too much and you don't need the energy, you bring them into fat cells or adipose cells, and you store them. You store them until they're needed. That's what I want you to know so far. Couple more things that I want you to know, and then we're going to move on. So don't worry about all the complex biochem of everything. I want you to know that after the process of digestion by lipoprotein lipase, there are these things called chylomicron remnants. So they're just the product of digestion of a chylomicron. Chylomicron remnants, and there's some protein containing compounds. And these particular protein containing compounds also contain a little bit of lipid, and there are various types of we call them lipoproteins. So you get chylomicron remnants and you get various lipoproteins. And these lipoproteins are named based on how dense they are. The more protein they have, the more dense they are. So if you have a very highly dense lipoprotein, we just call it an HDL or high density lipoprotein. If it doesn't contain a lot of protein and it contains mostly fat, we call it a low density lipoprotein or LDL. And this is where the term cholesterol comes from. So when people are told that they have high cholesterol, we're not specifically talking about that cholesterol molecule that we've been discussing. We're specifically talking about these lipoproteins. And specifically, the one that's bad in the form of high cholesterol is the low density lipoprotein. And I'll talk very briefly about what this means. Both of these products are metabolized further in the liver. That's all I want you to know. I don't need you to know any of this other stuff here. So for these past two slides, if you're feeling overwhelmed with what's on the slide, I want you to know only what's written at the bottom of the page, these 3 points. And I want you to understand where things are coming from for these 3 points. And I want you to know these 2 points for this subsequent slide. The rest of it, I don't need you to know. I don't need you to know about lipoprotein complexes and the metabolism of things and bile salts being returned. I don't need you to know about that. Okay, so as I mentioned, lipoproteins in the body, they're made in the liver. And the heavier the lipoprotein is, or the more dense it is, the better it is for you. So there's specific types. There's very low density lipoprotein that contains mostly fat. There's low density lipoprotein that contains still a low percentage of protein and a high percentage of fat. And then there's high density lipoprotein or HDL. When we talk about high cholesterol in the body, we actually talk about the LDL. If the LDL level is high, that's considered bad. If you have a high HDL, that's good cholesterol. That's fine. We don't worry about high levels of HDL in the blood. And the reason that we think about low density lipoprotein as the bad cholesterol is because this low density lipoprotein is very toxic to the lining of the arteries, and the toxicity that it has to the lining of the arteries causes there to be a lot of inflammation, and it's very highly associated with narrowing of the arteries. And so with the plaque formation in the arteries, and that plaque formation, the process by which that occurs, it leads to a condition called atherosclerosis, which you've probably heard about. You may just not have recognized what causes it. We're going to talk a little bit more about atherosclerosis in the cardiovascular section of this course. So that ends our discussion on carbohydrates, proteins, and fats and how they're broken down and how they're absorbed. I'm going to have a brief discussion on some of the vitamin absorption. This is going to be not in as much depth as you'd get in a nutrition course by any means. I just want you to have an idea about how things are absorbed from a vitamin perspective. We're going to go through maybe 3 or 4 different varieties. So blurb on vitamin absorption. The fat-soluble vitamins are the easiest ones to understand. Fat-soluble vitamins are vitamin A, vitamin D, vitamin E, and vitamin K. These are in alphabetical order. I don't remember them in alphabetical order. I remember the acronym CADE, K-A-D-E, and that's how I remember. Those are the fat-soluble ones. Everything else is water-soluble. And this is pretty easy because the fat-soluble vitamins are absorbed in the small intestine in the same format that fats are absorbed. So these things just get packaged in those little micelles, in the core of those micelles, and then get absorbed in the small intestine. There are some fake fats out there. So olestra, I don't think it can be used anymore anywhere, but it's definitely not in Canada. It's a fake fat that cannot be absorbed. So you can ingest foods that have fat in them, and that fat is just not absorbed at all. It just passes right through. But the problem is that it also very highly inhibits the absorption of fat-soluble vitamins. So these fake fats like olestra can cause you to become very deficient in the fat-soluble vitamins. So that's a bad thing. Water-soluble vitamins, when we think water-soluble, we think of vitamin C and all of the B vitamins. There's lots of B vitamins. They're absorbed, because they're water-soluble, they are absorbed via transport. It's a facilitated transport that has to occur in most cases. This is except for vitamin B12. Vitamin B12 is an interesting one, and I want to talk about vitamin B12 because it is such an interesting vitamin. The only place that vitamin B12 is absorbed, there's only one location, and it's in the very end of the small intestine. They refer to this area as the terminal ileum. That's the only place it can be absorbed. Anywhere else in the small intestine, you're not getting vitamin B12 absorption. So if somebody had surgery and had their terminal ileum removed, they can't absorb vitamin B12. That's plain and simple, you can't absorb it. So a person who has had their terminal ileum removed, you have to give them vitamin B12 injections so that it goes from the muscle directly into the bloodstream. The vitamin B12 also has to be, it has to be attached with a specific protein. And this specific protein is called intrinsic factor. If it's not attached to this intrinsic factor, it cannot be absorbed. So vitamin B12 is absorbed in the terminal ileum and only when it is attached to this thing called intrinsic factor. And the intrinsic factor actually comes from a cell type that we've already talked about, the parietal cells. The parietal cells of the stomach, the exact same ones that release hydrogen ions or protons for hydrochloric acid secretion, these parietal cells also make intrinsic factor. So it's released in the stomach. And so any B12 that you're ingesting, once it's in the stomach, The parietal cells are making that intrinsic factor. The intrinsic factor is binding onto the B12, and it travels through the small intestine. And once it gets to the terminal ileum, it can be absorbed. If you don't make intrinsic factor for some reason, or you don't have a terminal ileum, you can forget it. There won't be any vitamin B12 absorption. And B12 is an important vitamin. It's an important vitamin because it is responsible for normal red blood cell development. People who don't have enough vitamin B12, their red blood cells don't form normally. And if you actually take a sample of the blood and you look at the red blood cells under a microscope slide, the red blood cells are large, they're kind of strange looking, and they don't carry as much oxygen, they don't have as much hemoglobin, and you don't make as many of them either. So you have a low number of red blood cells. And people who are deficient in vitamin B12, they end up with a condition called anemia. And there's lots of reasons why somebody can become anemic. You can either have not enough iron so that you're not making enough hemoglobin, or you can not have the vitamin B12 and you don't develop your red blood cells normally. So you have a low number of red blood cells. Anemia, we break down the word, emia means blood, and the prefix with the letter A or an, it just means lack of. And so lack of blood means you have low blood. This is always referring to red blood cells though. And so you can imagine what might happen if somebody has low red blood cells. You can't carry as much oxygen. Your cells aren't getting the right energy that they need, or the right amount of oxygen, so they can't make as much energy. So somebody with anemia often feels fatigued, often has low energy, might be sleepy. They might feel dizzy when they're walking. If they're trying to get some exercise, they might almost pass out while they're exercising. And so a person who is deficient in B12 may have some of these symptoms of anemia because their red blood cells aren't developing as they should be. So interestingly enough, a person, if say that they're for some reason aren't making intrinsic factor, you can't replace it. If you don't have a terminal ileum, you can't absorb vitamin B12. So the treatment for a vitamin B12 deficiency is giving injections of vitamin B12 into the muscle. So they call that within the muscle, intramuscular shots of vitamin B12. Talk slightly about iron digestion. So iron is a substance that is ingested in two forms. It can either be in the form of heme iron. So this is actually the type of iron that we get from animal products, from hemoglobin in animal products. And you can see that this heme iron is actually surrounded by this ring structure. And the iron atom, or the iron ion, is at the center of this ring structure. So that's the first form that iron can be ingested. The second form is actually in ionized form. You can actually ingest ionized iron. It's either as an Fe2 plus form, so with a valence of +2, or it can be ingested in Fe3 plus form. The thing to remember is that you can absorb heme iron, you can absorb Fe2 plus iron, but you cannot absorb Fe3 plus iron. So you can ingest it all you want, it won't be absorbed. You have to actually change the valence of this to Fe2 plus before it can be absorbed. If you don't do that, it passes right through and you won't absorb it. A lot of plant-based iron is Fe3 plus iron compared to Fe2 plus iron. So plant-based iron is not as absorbable as animal-based iron. And so here's how iron is absorbed. So here's the lumen of the small intestine. Here's the intestinal cells. And then beyond here, you can imagine that there would be a capillary for iron to be absorbed. So the first way that we absorb iron, you can absorb heme-based iron. And there's actually a heme transporter. If you have heme iron, you will absorb it directly into the cell with the heme transporter. Once the heme is in the cell, it gets broken up into the actual iron atom, or iron ion, sorry, Fe2 plus, and that ring structure. And that ring structure is just called the porphyrin ring. So this is the porphyrin ring structure of heme. And so once it's in the cell, the ion gets removed and you're left with the porphyrin ring structure. That porphyrin ring structure is then broken down and degraded. The second way that we actually absorb iron is via the ionized form. So we can have Fe2 plus iron. It's taken up into the cell and it is brought into the cell with a co-transporter with a proton. That Fe2 plus iron is then brought into the bloodstream and it's allowed to go into the bloodstream with another transporter called ferroportin. So it kind of ports the iron into the bloodstream. The ionized form here where we absorbed, where we're starting the process of absorption of the ionized form of iron, this particular transporter is called, it's such a not so creative name. The transporter name is the divalent metal transporter 1. So there's multiple types of divalent metal transporters, but it's called divalent because there's a charge of +2. It's a divalent ion. Metal, because iron is a metal, and it's a transporter. That's the only reason why it's called divalent metal transporter 1. And then again, that ionized form of iron, whether it comes from heme or it comes from the diet itself, it is then brought into the bloodstream by the help of that transporter called ferroportin. Question is, what happens if we ingest the Fe3 plus iron? There's no transporter that will help the absorption of the Fe3 plus iron. Fe3 plus iron, there's actually a nutritional trick that you can use to, if you eat a lot of plant-based material that might have some iron in it, there's a trick you can use to help convert the Fe3 plus iron into the Fe2 plus iron. And the trick is that, and I'll give the example of oat products have a lot of iron in them. And this iron is in the form of, it's the Fe3 plus form of iron. So if you're eating something like oatmeal or granola or something, at the same time, ingest that have orange juice or some sort of an acidic juice or a citrus juice, and that will help you convert the Fe3 plus iron into Fe2 plus iron so that you could absorb more of it. It won't convert all of it, but it will aid in the absorption. The only point to make about that is that you have to have the citrus juice at the exact same meal that you're eating the granola or the oatmeal. Otherwise, it won't work. It has to be on the exact same meal. You can't say, "Oh, I ate oatmeal this morning. I better drink some orange juice." You're too late. So that's iron absorption. The last one we'll talk about is calcium absorption. And calcium absorption is very highly regulated by vitamin D. So anytime you think of calcium absorption, think of vitamin D. Some calcium is absorbed in an unregulated fashion between the cells, but this is the minority of the calcium. Not a lot of calcium is done via this mechanism. It's mostly vitamin D. And so we'll just quickly look at how calcium is absorbed. Again, the left side is the luminal side. The right side is the side with the capillary. And so The first thing, calcium will enter the small intestinal cell or the enterocyte. It'll enter the enterocyte um, by calcium channels. Okay, so the calcium goes in and it's actively absorbed. It's either actively absorbed by a calcium pump or it's actively brought into the bloodstream by a sodium calcium exchanger or an antiporter. This process is regulated by vitamin D. The more vitamin D you have, the more of an ability you have to absorb the calcium into the bloodstream. The secondary method is this method whereby you have calcium that sort of goes between the cells. And this particular type of calcium absorption is called the paracellular absorption method. It's not regulated, and it doesn't occur in high enough quantities to allow for you to have an adequate calcium level. So the process by which it is actively absorbed by going through the calcium channel and then pumped into the capillary is the form that gives us the most amount of calcium, or allows for the most amount of calcium absorption. And that process is regulated by vitamin D3, not vitamin D2, it's vitamin D3. I almost always ask a multiple choice question on this, so expect one. So here's just an overview of what we talked about. Well, we talked about a lot of these. We didn't talk about all of them, but just where things are absorbed. And so the esophagus absorbs nothing. It's just a vessel for food stuff to enter the stomach. In the stomach, there's actually a little bit of absorption that happens in the stomach. We didn't talk about it, but a little bit of water can be absorbed in the stomach. Alcohol can be absorbed in the stomach. Copper can be absorbed in the stomach. By far, the most absorption that takes place is in the small intestine. Everything from our monosaccharides, amino acids, and lipids to almost all of our B vitamins, almost all of our vitamins, almost all of our minerals are absorbed in the small intestine. There's the lonely vitamin B12 that is absorbed right at the end of the ileum. And the thing that we didn't really talk about is what's absorbed in the colon. What I want you to know about that is that the basic ions like sodium, potassium, and chloride, we also call those electrolytes, the basic electrolytes. So basic electrolytes are absorbed in the colon. Water is absorbed in the colon. And a couple of vitamins are also absorbed in the colon. Vitamin K, very important for clotting, can be absorbed in the colon. And this molecule called biotin, it's a B vitamin, also absorbed in the colon. So you might be thinking, why do we have vitamin K twice here? So most vitamin K is ingested from the diet and brought into the small intestine, absorbed in the small intestine. The reason why we have a little bit of absorption of vitamin K in the large intestine and a little bit of biotin absorption in the large intestine is because the bacteria within the colon produce vitamin K and produce biotin. So while you may not have even ingested vitamin K or biotin, the bacteria in your gut produce it. And so you will absorb some of that in the large intestine. So I recognize that was a lot of material. You may need to go back and really understand the processes. I like to ask questions about processes. So I like to ask questions about the process of single amino acid absorption, or the process of digestion of carbohydrates, the process of. So if there's any stepwise processes that we talked about, I recommend that you know those really well, because they will help you in answering some of the questions on your exam. In this particular installment of physiology, the GI physiology. There's one more installment where we're going to talk about the regulation of um, digestion and the phases of digestion. Um, and after that particular presentation, that's what's going to be covered on your first midterm. So while we might get started, we will get started on the cardiovascular section. We're not going to cover the cardiovascular section on your first midterm. So the things that you need to know for your first midterm are going to be the metabolism, all the basic biochemistry, metabolism, and GI phys.